Good afternoon. Welcome to the Royal College of Pathologists World Antimicrobial Awareness webinar. My name is Peter Johnston. I'm one of the vice presidents at the college. I'm VP for Professionalism, and I'm delighted to be hosting this webinar this afternoon. We have a panel of experts from a variety of specialties. The first is Dr. David Jenkins, who is a microbiologist, Bernie Croal, a chemical pathologist, Patrick Yong, who is an immunologist, Golda Shelley-Fraser, who's a medical examiner, Simon Stern, a haematologist. We've also got Arlene Brailey, who is a Liaison officer in Antibiotic Research UK, Fergus Allerton, Fergus, who is a veterinary pathologist, Mervyn from intensive care medicine, and Simon Stockley, who is a general practitioner. So from this group of specialties, what we will be asking is that they provide us with insights from their specialties perspective and how antimicrobial resistance affects their practice and their patients and the work they do. We'd like to address the challenges of antimicrobial resistance as we see them, and also the approaches to AMR that we might adopt in order to try to minimize or prevent harm to patients. How this will work is that, first of all, David will give a brief introduction, followed by a panel discussion for about 20 minutes, followed for the next 25 or 30 with questions from the audience. In order to have these questions considered, if you could use the Q&A function, that would be very helpful. And if you give us the questions, we will try to put these to the panel on your behalf. The audience will remain muted during the course of this. So thank you very much for coming. Thanks in advance to the panel for their participation. And I would like now to pass over to David to allow him to give a brief introduction to antimicrobial resistance. Thank you very much, David.
Well, many thanks, Peter, and thanks for the introduction and for the invitation to join in this discussion. It's obviously very apt to talk about antimicrobial resistance in World Antibiotic Awareness Week. Antibiotics are something that drugs that were first used in the early 1940s, following the discovery of penicillin by Alexander Fleming in 1928-29. And over the last 80 years or so, we've seen both a flourishing in the array of antimicrobials available for treatment, treating a complete range of bacterial infections that cause human disease. But also we've seen the increasing fight back by microorganisms, particularly bacteria, but also fungi and viruses, against the action of antimicrobials such as antibiotics. And this is really a very important and urgent problem to address, because it's often been said we face the prospect of a post-antibiotic era where antibiotics no longer work. But actually, we're probably already in the middle of that, because there are many parts of the world where antimicrobial resistance to a wide range of very important pathogens is already in place, causing problems, causing deaths as a consequence. And a recent paper published this year carried out an estimate of the number of deaths that RNA came up with is 1.3 million people worldwide. Most of these deaths are in probably the poorest areas of the world, low middle income countries, and that's something to be aware of. But we also know that antimicrobial resistance can spread. It can result of resistance genes moving from bacteria to bacteria, but also spread by resistant bacteria moving from person to person. And there's also, of course, people carrying resistant bacteria around the world. And we already see in this country, in the United Kingdom, outbreaks of antimicrobial resistance, which are very difficult to deal with. They can cause outbreaks in hospital settings. They can be very difficult to treat. Patients die as a consequence of them. And a number of these outbreaks have their origins in low middle income countries. So it really is an issue that we should all be aware of, we should all be concerned about, and we should all be doing something to tackle resistance. Now, I've talked about resistance for quite a bit, where I haven't actually said what it is yet. So resistance is the ability of microorganisms, particularly bacteria, to withstand the killing effect of antimicrobial drugs. And these resistant bacteria, resistant microorganisms are different from their sensitive counterparts because they have molecular structures or biochemical differences that make them less susceptible to the killing effect of antimicrobials. And fundamentally, this is down to genetic selection of these organisms. Resistance is due to random mutation of genes or transfer of resistance genes, and that's something which is particularly challenging. Bacteria can swap genes one to the other, and so resistance genes that are successful and help bacteria to survive get swapped around most frequently. And so success leads to success, at least from a bacterial perspective. The other thing to bear in mind is that although antimicrobials have only really been in clinical practice since around 1940s, 1945 or so, in fact, antimicrobials are ancient. Penicillin itself was derived from a mold, a penicillium mold. And this is a mold that's been producing this penicillium compound for probably hundreds of thousands, maybe even millions of years. And so as a consequence, environmental bacteria, environmental microorganisms have had millions of years to develop resistance mechanisms. So both antimicrobials and antimicrobial resistance is ancient, predating human use of antimicrobials. But what's happened over the last 80 years or so is that the use of antimicrobials in clinical and veterinary practice, including animal husbandry and also agriculture, has really turbocharged the development of resistance. And so we've seen a massive increase in the amount of resistance that's developing, often hand in hand with the introduction of new classes of antibiotics coming into clinical practice. Now, the problem is that whilst in the middle decades of the 20th century, it was a bit of a golden age with lots of new antibacterial drugs being discovered and introduced into clinical practice, over the last 20, 30 years, antimicrobial development has really dwindled to virtually nothing at all. In fact, the last new antibiotic class to enter clinical practice was discovered back in 1987. And so we really are dealing with very few new agents. We do see some new antibiotics being licensed, being introduced, but these are new representatives of old classes and don't represent that kind of massive breakthrough that we really want to see. So the question then is, how do we deal with the situation where antimicrobial resistance is increasing, becoming more widespread, not only to new antimicrobials, but geographically wider as well, whilst at the same time not having access to new antimicrobials to tackle this resistance? We're really facing a situation where antibiotic effectiveness is dwindling year on year, and we really need to tackle that and do something about it. And it'd be very, I think this will be a really interesting discussion, and I'd love to hear ideas of what people have, how antimicrobial resistance is affecting their specialty clinical practice, but also what they're doing about it and what they want to do about it. So over to you, back to you, Peter. Thank you very much.
Thank you very much, David. Well, seeing as you asked that question, I think that's the one to pick up on, isn't it? So if we just go around the room, I think we could have cameras on for the panel, please. If we go around the room, we can ask how it affects our individual specialties. I guess if we think about patient journeys and the influence of diagnostics on those journeys, it might be wise to start with primary care and Simon Stockley, who sees the patients first and then progress through the system, through the diagnostics to come to Mervyn around intensive care medicine. So that we have a story to tell as we go. And others may wish to join in coming along, but and then I think after that we can move on to talking about the science behind where we are and what we can do scientifically to improve things. So if I can start with you, Simon Stockley. Sorry, we've got two Simons, which is why I'm saying Simon Stockley and not Simon Stern, but Simon, what I can't even say Simon S because they're both. So, Simon, how would you respond to David's question about how does antimicrobial resistance affect your practice in primary care?
I think the first thing to say is it has changed significantly over the 30 years that I've been in practice, from when the last antibiotic was introduced to the current day. The approach to managing infectious disease has been far less just in case medicine. I'll give you an antibiotic just in case it will make a difference, and an expectation of antibiotics for febrile illnesses to ones where we are seeking out an accurate diagnosis. We've had a sea change in the last 2 years with the advent of lateral flow tests and PCR testing for one particular infectious disease, but others have been around for a little while, and we're beginning to feel more confident in the conversations we can have with patients around their infection. The counterbalance to that has been the increased awareness of sepsis and the desire not to miss the very serious illness. And a lot of my work has been on persuading GPs that they do have the skills with which to be able to safely manage a febrile illness without falling into the trap of having to give an antibiotic. And we've become very much more selective about the antibiotics we use and have stopped using blundicillin for every infection, because then we didn't actually have to know an awful lot of microbiology, because we're pretty sure that coamoxiclav was a wonder drug and we could use it safely. But now we know that carries with it significant costs for the patient and for society. So I think things have changed, but we still prescribe a little bit just in case and fear of missing something serious.
Thanks very much. I think one of the questions that is interesting in this is a bit about the point of care testing that lateral flows have brought us for COVID. I don't know whether any of you would like to comment on those sorts of diagnostics as regards antimicrobial therapy in general. I mean, one of the difficulties of empiric prescribing, which we've all recognized, is the fact that when you're a GP, you don't have the opportunity to wait 3 days just as intensive care. We've had this discussion too in the past, Mervyn, that you can't really wait for 2 days to deliver a diagnosis that's going to be the thing that you need to be specific about your treatment. And so I guess, how can we smarten up, if you like, our response to the clinical situation where we really need a diagnosis now, if we're going to be able to do that? I mean, I know you've done work on point of care testing before, Bernie, with stuff. I just wonder if you've got any insights into the use of point of care in terms of a microbial diagnosis other than COVID.
Yeah, thanks, Peter. I mean, certainly we're beginning to see a rapid expansion in the number of different types of point of care testing. You know, so the testing that's really exploded around viruses and even bacteria, you know, is useful in that setting. Obviously, I'll leave it to the microbiologist to comment on that. In terms of biochemistry, there has been a sort of an attempt to look at, say, CRP testing, for example, in primary care as a way of helping indicate whether or not there is you know, a bacterial infection in need antibiotics. And in some ways, although the test isn't perfect, it's at least acting as a sort of way to triage patients, and you know, that's possibly something that is of use. The difficulty, however, like all point of care testing, is implementation, safe practice, and who's going to pay for it. You know, and that is always the stumbling block, and there's a danger that we end up with, you know, patchy implementation, and also you know, patchy safe practice in terms of how the point of care testing is actually used, monitored, and, you know, quality controlled. So but I think, you know, as time moves on, we're beginning to look at existing tests and new tests such as procalcitonin, you know, as potential ways to assist in the whole, you know, dilemma of trying to establish whether there's a significant bacterial infection present that could benefit from antibiotics. But as with everything, there's a cost to it.
Thanks. I'm just thinking about, you know, how this is applicable in the community. And before I come to the hospitals, I wonder, Fergus, if we've mentioned the interaction between animal and agriculture populations in relation to antimicrobial resistance. I wonder if you've got a comment at this point.
I think we share a lot of the same difficulties that are seen in medical practice, and probably we can consider ourselves a few years behind medicine in that there is an increasing move to look for an underlying bacterial pathogen when we wish to use antibiotics. But I think the default is still just in case prescribing, and this partly reflects the discordance between costs, and as we are representing private healthcare, there is no National Health Service for pets. We are seeing that owners would rather have that defensive coverage of antibiotics than look at expensive testing to try and achieve a diagnosis and identify the causative agent. So we are having very similar challenges, and I think we are very keen to engage all our pet owners in this and try and encourage them to be aware of the importance of antimicrobial resistance, and also for vets to take a very careful, prudent, rational use to antibiotic use. But
thank you. I think, no, thanks very much, Fergus. I think it's interesting that the parallels are what you say, the similarities. At the same time, I get the pressures that you're under to do things, and I think probably you know, that there is a pressure across medical practice in general to be seen to be doing something, and the idea of go way back and come back in 3 weeks, and if you've still got a sore throat, I might think about it. It's difficult, you know, for anybody to deal with, but it's interesting, and thank you for that. I guess it's thinking about the value to the community as a whole rather than to the cost or value to the individual, which is a difficult question, a sort of ethical question, which we might come back to later on. Moving towards the kind of hospital side of things, it strikes me that, Patrick, as an immunologist, you often see immunologically compromised patients, and Simon, as a haematologist, some patients are immune compromised because of the disease, but also because of what we do to them to cure their diseases. So I guess I wonder if one or other of you would like to come first about this and say how you feel antimicrobial resistance is affecting your practice at the minute. Simon, I think.
I can think of perhaps 2 incidences in the last year when we've actually found it very difficult to find an antibiotic for one of our septic inpatients. So it is a real issue. And in some ways, we're at the other end of the clinical spectrum to the other Simon S in primary care, because the one thing we drum into our SHOs when they start with the team is, you know, you can't hang around if you've got a neutropenic patient who's febrile. So we, as all haematology units do, we have empirical protocols for what we do in that situation. And that, of course, involves broad spectrum antibiotics to cover as many organisms as possible. And, you know, that has its upside, but also the downside, in that we only I mean, our local audit showed we only get positive blood cultures in 30% of patients. Well, I think that replicates sort of national published or international published data, which means that patients stay on these empirical broad spectrum drugs for prolonged periods of time, up to a week. And that's not, in terms of avoiding antimicrobial resistance, can maybe thought not to be best practice, but we are operating without a clear-cut microbiological diagnosis in that situation. So what do we do to try and minimize problems? Well, antibiotic stewardship's the buzz phrase, but there's a lot to be said for it. A lot of pharmacists checking that patients aren't staying on certain antibiotics too long, good handovers between the medical teams, say at weekends, just to make sure that if antibiotics aren't providing the desired clinical effect, they're changed, and we don't just keep on and on. So it's a situation that can be managed, but it's not ideal. And I think what a lot of us in various specialties would say that would be the future, I hope, would be improved, maybe molecular genetic diagnostics that will allow a quick diagnosis to be made without necessarily waiting for the prolonged cultures that we now out of necessity have to wait for.
Thank you. Patrick?
So very similar to Simon's answer, we see patients with immune deficiency as well, who have difficulties clearing infection because their immune systems don't work as well. So antimicrobial resistance is a big problem if you don't have any antibiotics to use. And the converse is true as well. And that one wouldn't wait too long to start antibiotic treatment. Rather than waiting for cultures, you would go on a just-in-case basis, because infection is obviously more dangerous in persons immunocompromised, immunodeficient.
Thank you very much. Just moving along the patient journey, Mervyn, as an intensivist, how does this affect you?
Yeah, thanks, Peter. Well, in two ways. From a patient point of view, and there are lots of studies to show that you don't need to rush and throw antibiotics at anything that moves. And clearly, you take hopefully a measured approach. If they're very sick, your response time should be shorter than if they're sort of in inverted commas less sick or mildly sick, and so I think now it's accepted that you do have a window of opportunity to actually get some tests done to actually first work out, is there an infection? If there is it bacterial, fungal, viral, atypical, etc.? And then hopefully, if it is bacterial, to try and get the right antibiotic first time, because I think the evidence is more compelling that giving appropriate empiric first-line antibiotic therapy is much better in terms of patient outcome. So we want to get it right first time. And unfortunately, as David mentioned in his talk, there's it's happening in the UK, but thankfully to a lesser extent than is happening in Southern Europe, Asia, South America, North America, etc., Africa. So it will no doubt catch up with us eventually. So we're behind the curve, which is good to be behind the curve for once. But at the same time, it is ominous that there is a year-on-year increase. So ideally, we need to, as has been said by lots of other people, restrict antibiotics where they're not needed. For example, Simon mentioned, over-treating, and certainly lots of studies, for example, with community-acquired pneumonia that require hospitalization, the vast majority are viral. And so clearly, point of care testing, to my mind, is the answer, because we don't want to wait 1, 2, 3 days or whatever for a result. And there are thankfully lots of companies out there making a whole variety of molecular diagnostics that will give an answer quickly, even at the bedside. And this is for blood, urine, CSF, stool, bronchoalveolar lavage, etc. And for example, we were using, I won't mention the company's name, but we were using one such kit during COVID as a way of not giving antibiotics to patients. There was obviously about a 3% co-infection rate with the SARS-CoV-2 virus and bacteria, but And clearly patients who are sick in intensive care units are prone to getting secondary ventilator-acquired pneumonias, but it gave us the additional comfort blanket to not throw in antibiotics if there was no evidence of a bacterial or a fungal pathogen. And these panels now can give you 20-odd different pathogens and some of the common resistance genes. So it's not catch-all, it's not 100% perfect, but I think it's a sign of things to come, that this sort of rapid technology is coming, and it'll be on our doorstep now and improving very quickly in the next few years. As was mentioned earlier, cost will be an issue, but I think you can justify the cost with hopefully a good health economic analysis to show that actually getting the answer quickly, giving the right treatment, saves time in hospital, saves lives.
Thank you. Thanks very much for that. Before asking Arlene if there are research questions or research solutions that are ongoing, one of the things that we've spoken about earlier, Golda, is the importance of having awareness in the public as to what's going on. And we've discussed various aspects of how patients are cared for. But from the point of view of a medical examiner, what are the key issues as you see this going forward?
Thanks, Peter. So, infectious diseases and sepsis are still extremely common causes of death worldwide, obviously more so in developing countries, but still very much so in developed countries, and especially amongst the elderly. And so as medical examiners, we're very much focused on accuracy of death certification, and also on exploring to ensure that accuracy, exploring whether we thought there was a risk of antimicrobial resistance, and if that's detected, passing that information on to the bereaved. So for us as medical examiners, when discussing with certifying doctors causes of death and infectious diseases in particular, we'd speak about antibiotic regimes. Obviously, we would have a different amount of evidence depending whether it's a community death or hospital death. So for those patients in the hospital who may have had cultures and less broad spectrum antibiotics, it might be a different story to those in the community. Discussing with those certifying doctors, or if necessary, microbiologists or pharmacists, whether there was any suspicion of antimicrobial resistance, if that was a factor that we think was contributory in the death. And if so, recognizing that on the death certificate. And then when we speak to the bereaved about the wording on that certificate and asking them if they had any questions about their loved one's care in their final stages of illness, that could be something that we raise. So it's about incorporating the issue into our discussions for those deaths around infectious diseases.
Thank you very much. I think that's really helpful. And I think looking at all the kind of summarizing what's come up and looking pointedly at Arlene, the topics for research as I see them out of this are the epidemiology of what's going on, the idea about developing new antimicrobials, and how we develop diagnostics in order to help link it all together. So I wonder, what have you got going, Arlene, that's of relevance, and how do you think how are we addressing these issues with research?
Thanks very much, Peter. Well, of course, I represent a charity called Antibiotic Research UK, where there is a research programme working across, the academia, pharmaceutical industry, etc. My own particular role, though, is to lead the patient support programme, which is about supporting and listening and signposting those with resistant infections, those with ongoing recurring infections. So maybe I could summarise it by saying these are the things I've learned as I've listened to them. So, the first thing that patients talk about is language as a barrier at the beginning. So they don't use the same language as we do as health professionals. There's not enough communication with the patients who've been diagnosed with these infections, so they say, and not enough information given, and I would use the example of Sharon, who told me, she phoned me in a panic saying, I've just been discharged from hospital, told that I've got an ESBL. Nobody could explain to me what that was. And I don't know if I can hug my family. Can I cook for them? Can I share towels? You know, can we live together as we did? An absolute panic. So there's something about appropriate information, appropriate support. And I could go on to tell you a little more if we've got time later about what happened when she was readmitted in other occasions. Patients will talk about the whole issue of drug resistance is just not understood until it's experienced by them or a member of their family. So, you know, I think they would say the public don't get it till they get it. So we've got a lot of work to do, I think, to cross that bridge. And of course, they all have the same concern, which is that worry that there'll be no antibiotic left to be able to treat them. They talk about living with fear and dread, the dread of a recurring infection or a resistant infection flaring up again, and all that involves in terms of the experience, but also the fear that they're going to be told one day that there is nothing else left to use. Quite a few I speak to have been told, you know, they're down to the last 1 or 2 antibiotics. That's quite a scary message to hear, isn't it? And I suppose the other aspect to bring in is the fact it doesn't just affect them physically. I've learned that these infections affect their whole life. So they affect their social life, they affect their ability to work. Many lose their jobs, their relationships, and importantly, their mental health. And I think that's a big safety issue that we might have overlooked, that actually a lot of these patients talk about not wanting to live like this. And so there's some serious issues in there too. And I think finally, just to say, an area we've maybe not looked into is the fact that a lot of these patients feel they're not believed by family and friends because it's an invisible illness, isn't it? They can look perfectly well, but they're carrying resistant organisms. They have recurring infections, whatever it might be. And they talk about the fact that they don't feel believed. And sometimes that's true. They say of health professionals, because the diagnostic testing says nothing found, therefore you don't have an infection, all is well. They know that they're really unwell. So lots of research areas I think we need to cover in all of that, you know, beginning with the diagnostics, as others have said, and taking it right through to, have we got new treatments on the way? Are there new combinations? Are there non-antibiotic therapies such as phage and vaccines, which these patients are investigating for themselves in desperation?
Thank you very much for those insights. Just sort of ties it all together beautifully. Thank you. I think we should move on. Thank you all for your comments around David's first question. Looking at the questions that have appeared as we've been talking, there are a couple around about costs and how do we manage to balance the notion that we want to increase increase everything, really. We want to be better informed, we want to know more, we want to diagnose quicker, better, faster, we want to treat more effectively. How do we balance the cost and the workload, given the fact that, you know, people are not entirely twiddling their thumbs to pass the day? How do we manage to get more work done? I guess it's a question of effectiveness over cost and the fact that we get benefit out of it. But what are the processes that people see about delivering what really is a better system, a safer system in which antimicrobial use continues to be regulated in a way, or at least overseen, but how do we make that stewardship more effective? And so I don't know who wants to start with that one, but I don't know. David, do I point it at you?
This is a problem I've been trying to deal with for a long time. So I was hoping that someone might be able to give me the answers here. But I mean, stewardship is something which is increasing across the UK, across the world. In fact, apart from being a medical microbiologist, I'm the current president of the British Society for Antimicrobial Chemotherapy. It's a British organisation, but one with a global perspective, and it's something that we've introduced just recently is the Global Antimicrobial Stewardship Accreditation Scheme, GAMSAS, which is an attempt to identify good stewardship practice, but also identify where things could be better. And I think if we look at stewardship in this country, it's still, to be honest, it's still a fairly nascent subject. It's something which is carried out by a couple of people, generally speaking, antimicrobial pharmacists and a microbiologist and maybe an infectious disease physician. And I think they struggle a lot to really make a mark on hospital antibiotic prescribing. And it's even less well developed in primary care, although from what Simon was saying, actually the GPs have been doing quite a bit, and I think variably successful there. So part of the problem, I think, is trying to attack the issue of causing harm. I think people are very concerned that by restricting antibiotic prescribing, we will be removing effective treatment from people who might otherwise either die or have prolonged or more severe illness as a consequence. And I think part of the response to that is education, because as Mervyn was pointing out, a lot of people who get antibiotics for things like COVID, pneumonia, do not have bacteria causing an infection problem. And so we do really need to try and tackle that at various earliest level, medical school, of course. Although there's been a call to reduce antibiotic prescribing, in fact, at the same time, we're increasing the number of people, number of specialties, number of clinical practitioners who are allowed to antibiotics. And whilst that is good in many ways, including improving speed of treatment, it's on the other hand, it can, at least in theory, lead to more inappropriate prescribing. So I think the education element of antimicrobial stewardship is fundamental. But what we've seen in a number of different studies is that possibly the most effective, certainly the most immediate way of restricting antibiotic prescribing is through requiring prior authorization before antibiotics can be given. But that's very intensive in terms of resource. We introduced it in Leicester 14, 15 years ago when we were faced with a really big problem with Clostridium difficile infection. This is a bacterial gastroenteritis where antibiotic exposure is probably the #1 predisposing factor for this infection, which is a very serious infection with up to 20% mortality and associated with it. So no trivial thing at all.
Mervyn, would you like to?
Just to jump in. I'm well, perhaps a bit tyrannical, but I think antibiotics are often abused by junior doctors as a panacea. And I think you can quite gainfully tie the hands of the most junior, and therefore they have to actually seek agreement to start an antibiotic. And certainly what we do in our trust, which seems to be quite effective, is that after 4 days, they automatically get stopped by the electronic prescribing system. So it's got to be an active decision to continue them. So yes, hopefully a clinician should review at day 1, day 2, but even at day 4, then they have to make an active decision to continue the antibiotic course. So I think that's a good way of controlling use/abuse.
Oops, thank you very much. Simon Stern, you got your hand up to comment.
Just to echo what Mervyn was saying, we've got something similar at our trust, but I wanted to emphasize the key role. I mean, we're a very multidisciplinary panel, but one group we haven't got is pharmacists on the panel, and they're so important. And we were talking about costs and resources. What's really sad, and I don't know if other clinicians have seen this, is that when pharmacy is short-staffed, i.e. most of the time these days, sadly, due to the whole NHS staffing issue, very senior pharmacists get pulled from their stewardship role to sort of dispensing in the patient-facing pharmacy. And I mean, I'm delighted to hear that David is sort of really giving this national priority through his group, because I think, yet again, it's rather a false economy to be doing that. I understand why it's done, but it can leave the whole, we're a medium-large size acute trust without any pharmacist performing the stewardship role, so for a week at a time.
Thank you.
There have been quite a few things that GPs have been doing. pharmacists certainly have been with our prescribing and policing our use of broad spectrum antibiotics. I think one of the areas that we're perhaps a little bit weak on, it still is the number of people on long-term repeat antibiotics for somewhat dubious reasons. Often urology will start people on antibiotics for chronic cystitis, and you can find that they are still taking them months and years afterwards without a lot of review. And the same goes for skin conditions. I was very surprised in our own practice when we looked at the number of prescriptions that were going out was significant in terms of our antibiotic prescribing. It was actually repeat prescriptions which we weren't giving a lot of thought to.
Yeah, I think one of the messages that's coming out of this for me is that we kind of know what's happening, but we lack a really very good coordinated approach despite best efforts, and that coordination requires science, medicine, informatics, politicians principally too, to make sure it's all together. And that's about profile, isn't it? And we're not very good at that in most of our specialties about raising that profile. And I guess that would be something we could think about how we could do that. Specifically, I'll come back to you, David, on that, but there's a question that's come in about guidelines. And you've mentioned already, David, the idea of stewardship. So there is a question about the use of low-dose antibiotics, which guidelines from perhaps the likes of NICE might be encouraging us to use, whereas that might not be ideal, and therefore you're half-treating infections with the likelihood, therefore, of under-treating and helping the development of AMR.
I could perhaps just add here, Peter, that many of the patients I've spoken to who suffer from recurrent, maybe resistant UTIs have asked the question, is it possible that the short doses of antibiotics, 3-day courses, 5-day courses, repeatedly given to them because it hasn't eradicated it, could that have caused the bigger problem in the long run? And I think it's an interesting area for research to maybe follow through some of the patients later on that had short courses and perhaps have ended up with long-term recurrent or maybe even resistant infection.
Thank you very much. That's helpful for that. Patrick.
I'm just going to fill in the space while waiting for David to rejoin, but just one thing to add from my specialty's point of view, but essentially about antibiotic allergy as something else to look at in the area of antibiotic resistance. And the best example of this being penicillin allergy, which is a very common label, about 5 to 10% of the population, depending on which study, and 10 to 15 or 20% hospitalized patients. Most of the time, this label is usually inaccurate. Studies show me about 90-95%. And actually removing that label can actually result in better antimicrobial stewardship and sort of resistance. The BSAC, or British Society for Allergy and Clinical Immunology, has recently published guidelines on non-specialists. Essentially, we're like looking for patients with a so-called label penicillin allergy and removing it if appropriate. And I think David's back, and I've filled another minute there.
You know, I think that it's a very good point, and I think it's allergy. It's something that it is some. We've talked about this in another setting. I can't remember what it was, but I know I see your hand up too, Mervyn. I know that anesthesia, allergic to anesthetic agents, has also been something which keeps coming up. But if I can ask Simon Stockley first, then I'll come to you, Mervyn.
Yeah, I would completely support the view that penicillin allergy is completely overdiagnosed. I've been living the pain of allergy to COVID vaccines for the last 2 years, and actually true serious life-threatening allergy is remarkably rare to almost any drug, and the presence of a rash in association with an infectious illness has so long been labelled as penicillin allergy. Well, it was neither life-threatening nor was it allergy, and we've lost that ability to discriminate for these people, and we've not done them any service. So I strongly welcome people actually being just given penicillin as a diagnostic test of their allergy, if it's done sensibly with consent and understanding. The risk of actually doing somebody serious harm is, well, tiny.
Yeah, I think it was COVID, the COVID vaccine was the issue that Patrick and I have spoken about this before, that you brought to mind. Mervyn, do you want to come in now, and then I'll go to David to come back to the question that we started with.
Yeah, well, it was actually more on, pardon me, on duration of antibiotics. And in fact, we have to blame the Emperor Constantine, because he came out with the notion of a 7-day week. And had he come up with a 6-day week, we'd be giving 6-day courses. And so, unfortunately, it's just an empiric number that's been grasped out of the air. And increasingly, more and more studies have come through showing that, you know, for example, you know, Simon can comment, but urinary tract infection, 1 dose, a big dose of amoxicillin, you know, is adequate. Gonorrhea, again, 1 dose. You know, there are even studies from Africa with 1 dose for treating meningococcal meningitis. So we, unfortunately, traditionally have over-treated. And my personal view is we shouldn't be fixed with, or fixated, I should say, with a fixed dose or duration. But if the patient's clinically better, within a day or 2, you can stop, unless there's a deep-seated infection, osteomyelitis, endocarditis, that merits a longer course. And hopefully David will agree with me.
So Fergus, would you like to?
I was just going to comment on the shorter is better discussion, really, because we do believe in that in veterinary as well. I think Brad Spellberg's done a phenomenal amount of work on the medical side and shown these non-inferiority trials that shorter courses are achieving the same outcomes as longer courses of antibiotic for lots of conditions in people. We haven't got any of that evidence, and I think, as Arlene was saying earlier on, we don't know the impact of cutting these courses back down to a bare minimum. Will that maybe get temporary resolution but lead to greater recurrence? So we are trying to run studies. We've got a prospective multi-center study trying to look at different durations for managing sporadic cystitis in female dogs, and trying to establish whether 3 days is non-inferior to 7 days, both in terms of initial clinical outcome, but also in longer term recurrence. And I think we need those data, and I don't know whether dog data is going to be extrapolatable to people, but certainly it does seem to work the other way around. And I think hopefully if we can run some robust studies, we can demonstrate that shorter courses are as effective as their longer counterparts.
Yeah, thank you very much. I'm just looking to see there are some more questions that have arrived while we've been doing so. Thank you, audience, for keeping those coming in. One of the things is that's come up, which is valid and helpful in the comments, is about globalization of, or the global nature of the issue. And we've focused in this discussion principally around what's happening in the UK. And I think, you know, we've mentioned, it's been mentioned in passing about the fact that different areas of the world respond differently given their circumstances. And I guess the question is, how best can we engage with healthcare professionals and governments internationally to secure a way forward? And I guess this is not really about veterinary medicine or human medicine or anything else. It's a much more broadly based question. And we did mention earlier the ethics of all of this. And I wonder how that ties. What are your thoughts with all of that going forward? Anybody want to pick that one up? While you're thinking about it, for me, the notion is that we need a platform on which we can do something. And I guess the World Health Organization is one such. And I mean, there is an article that Lancet was tweeting about recently, which does demonstrate the variety of different antimicrobial resistance prevalence that there is across the world, with a series of interactive maps that the WHO is very good at. And I just wonder how we can better engage to make use of the data that they have, for example.
Yeah, I think it's an interesting dilemma, though, Peter, that, if you talk, when I talk to my colleagues in India, for example, because they have such a high resistance rate, they immediately jump in with either multiple different antibiotics all at once in the hope that one is effective, or the, in inverted commas, stronger antibiotics early on. And, they've painted themselves into a very desperate corner. And I think we can learn from that in the UK to try and minimize it. Yes, we have travel and people coming from abroad with multi-resistant organisms. But again, if we're cautious about how we approach it, and as Arlene mentioned, there are other strategies coming through, and also actually enhancement of existing antibiotics. There are ways of improving concentrations and kill rates of existing antibiotics. So I think, again, being behind the curve is actually, I think, quite beneficial for us. And yes, we have to learn from others' bad experiences.
Yeah, I think that it's really important to make sure we do that. I'm just thinking ahead about how, one of the problems we've got currently is we're confronted by a wicked problem, one that isn't going to get solved easily by making one or two changes here, but needs a much broader issue, much more broader approach. The difficulty of addressing these is, so whose job is it to do it? And we've currently been looking at this in relation to the new in vitro diagnostics regulations. Whose job is it to point out that we haven't really thought much about this and we probably need to? And so we are addressing that. And I wonder, Arlene, if you think, how do you think, because I think it would be helpful, how do we engage the patient voice in this, being able to use patients and the stories that the likes of which you have told to help engage with the media, with the politicians, to advance this as an issue where not only can we see its relevance to each of us as individual professionals, to specialties, but also to the global community.
Oh, it's like the Holy Grail in a way, isn't it? But I think the key thing is people like to hear about people, don't they? Our attention is caught when it affects one of our, human being partners, as it were. That's a poor phrase, but if something happens to one of our family, we're suddenly on alert. We want to do something. So patient stories are a way of putting context around some very difficult notions and some very difficult problems. I think what we need to do is harness that patient voice, use patient advocacy, and to help them to explain what it is that's happening from their point of view. So, for example, they can often see inconsistencies. If I go back to that story about Sharon, who had the ESBL diagnosis, she's been admitted to hospital a few times since on other issues. And she was put into a normal ward with other people. And she said, no, I need to be put in isolation. I carry this resistant gene. It happened that she was left in the main ward because the staff didn't understand the issue, and she was left petrified that she was going to be a risk to other patients, and she was also worried about herself and so on. So I think patients had a very strong voice to be able to know what should be done, what they can help to do. I think they also understand that, you know, the deeper issues around it. They know that diagnostic testing is a real problem because they've experienced it, and they can talk about that. They can also talk about the, you know, the experience of treatments where I'm thinking of a CF patient that spoke to me and said, the issue is when I'm admitted quickly to hospital in an emergency, I really need someone to get to that drug that's going to work for the kind of infection that I have. I know the IV drugs that don't work and haven't worked in the past, even though empirically they should. And so there's something about harnessing the expert patient in this, because many of them have learned to be an expert because they've really found themselves in such a place where nobody really has the answers.
Yeah, thank you very much for that. I think that's a really good lesson in there, Arlene, and one that we can all take on board, I hope. Simon Stockley has got your hand up. If you come in, then I'm going to come back to David in the hope that you're now reconnected, David. And you're still on mute, Simon.
Arlene raises a very important question as to how we manage infection control, both with the those who've got resistant bacteria, but also how good are we at putting immunocompromised and immunosuppressed people next to the patient with the difficult infection. I'm not sure that we can identify our immunosuppressed at the point of entry into hospital particularly well, and the chances of them sitting next to the old guy with bronchiectasis for a few hours is really high. We don't have any hospital data on hospital-acquired infections in immunosuppressed people because there is no data. There's no consistent way of picking up the immunosuppressed. I think we could do better with infection control.
Thank you. Arlene has put round a note on the chat, which you may be able to pick up, which is a reference to a patient leaflet developed by NHS Grampian, amazingly our own place, Bernie, on penicillin allergy and delabeling, and that is really very, very valuable because I think it's one of the real problems that faced for a whole range of different so-called allergies across the piece. I think we have reached the time of 2 o'clock, and we usually run the webinars for about 45 minutes. We said we would run this one a little longer, but I think 2 o'clock is probably time because people will have work to do in the afternoon. And so I would just like to draw some thoughts together out of this. I think what I'm getting is that antimicrobial resistance is a problem which affects us all as individuals, be we patients or not patients, but as people generally. It affects us through our relationships with animals and the environment within which we live and share with everybody. It's a problem that is global and that we require to consider how best to approach it. We've addressed things around about what would help us in terms of what things would be usefully know. And it strikes me that if we had some standardization of data gathering, it would feed well into stewardship, which would assist with managing this increasingly, I think dangerous, I think would be the word to use, position and threatening position as we go forward. So I guess questions are about how we collect these data and then what we do with the data when we've got them, to synthesize them into something which is useful for the prevention of making it worse, making antimicrobial resistance worse, but also helping us in the process of making best use of what we have available. And so when it comes to what have we got, we have science at our fingertips, albeit the challenges of not having had a new antibiotic since 1987 or a new class of antibiotics. there may be other ways, I guess, if we think of the different approaches to therapy for neoplasms, for example, that there may be ways in there perhaps which cleverer doctors than I would be able to explore. But I do think one of the real issues that we face is how we pull this together in the sight of the public and the politicians and the decision makers across the planet to enable people better to see the issues and to deal with them. So I think that we've reached the end of our time, and I would just like to go around the room and thank everybody for their contributions. Mervyn, Simon, both Simons, Arlene, Golda, Patrick, Bernie, Fergus, and David particularly. And I'm really sorry, David, we've had issues with the sound, because I'm aware of the fact that you're a fount of knowledge as regards what this issue is all about. And so I'm sorry about that, and there's nothing we can do about that going back, but thank you anyway, and thank you very much for the contributions that you gave us in advance to help us prepare for this. I would also like to thank the college team, Shane Johns, Maria Mary Rofeo, Gamze Zen, and Cynthia Mitri, who've put a lot of work into organising this webinar and running it to make sure we keep to script and keep to timetable. And thank you all for doing that. And thank the participants also for coming along, listening and posing your questions, which have been challenging and focused as ever. If you haven't had an opportunity, or you've got colleagues who haven't had a chance to see this or would like to look at it again, it will be available on the website in due course for you to come back. And just thank everybody for their participation in the day. And with that, wish you the very best for the rest of your day. Thank you.